Hello, my name is Leah Krubitzer, and today I would like to talk to you about combinatorial creatures, cortical plasticity within and across lifetimes. But I would first like to thank you for inviting me to give this really important presentation. And it makes me really happy that I'm giving a talk on animal research at a meeting on human brain mapping. So this is really exciting. Okay, so let's see here. Go. So today I'm going to focus on the neocortex. And this is a back view of a bottlenose dolphin brain, and this is a side view of a little Australian marsupial called the native cat. They're not drawn to scale, but you can see that there's a huge difference in the size of the cortical sheet in this bottlenose dolphin compared to this little marsupial. And why do I want to focus on the neocortex? Well, the neocortex is the portion of the brain that is involved in complex behaviors, including perception, cognition, and if you're human, language and reading. It's also the portion of the brain that's changed most dramatically in mammals over time compared to other brain parts like the spinal cord, the brainstem, and I think even the dorsal thalamus. So the big question in my laboratory is, how do you go from a very simple form that existed in our ancestral mammals some 200 million years ago? They had very few cortical fields, and each color represents a different cortical field, to the very complex cortex that a lot of extant or present day species have. And this is meant to represent a flattened view of the cortex, and all of these different colors are different cortical fields and modules within them. And I think there's some estimates that the human brain may have over 300 cortical fields. So how do you go from a simple form with maybe 15 or 16 cortical fields to a really complicated form with hundreds of cortical fields? Well, there's a problem. The evolution of the brain or the neocortex cannot be studied directly because brains do not fossilize, and modern brains are the results of thousands to billions of years of evolution. So I don't have like thousands or billions of years to sit around and study how this process occurs. But there are two ways we can circumvent the issue of evolution and understanding the evolution of brains. And the first is we can take a comparative approach. So we can study the brains of a lot of different species. And these are the products of the evolutionary process that occur over long time scales. And we can say, what changes has evolution produced? What are the similarities and what are the differences? And this is a really, really powerful approach. The problem is evolution is a moving picture of life. And even within a life of an individual, we are this moving picture of our own lives. And so when I take a comparative approach and I take that animal out of this moving picture of life, I'm studying a frozen frame. So while comparative studies can tell us what changes evolution has produced, they don't tell us how phenotypic transformations occur. How did some species get hundreds of cortical fields? We can take a developmental approach because the study of development is really, or evolution of the neocortex is actually the evolution of developmental mechanisms that give rise to some aspects of cortical organization. And this allows us to appreciate how changes have occurred, and we're looking at a shorter time scale rather than hundreds to billions of years. Okay, so what am I comparing within and across lifetimes? I can compare a lot of different things. What I'm comparing as a systems neuroscientist is functional organization, neural response properties, cortical connections, cortical architecture, gene expression, epigenetic marks, lifestyle, and behavior. So here's why the comparative approach is really powerful. And prior to the onset of non-invasive imaging in humans, we knew a lot about the brain. And I would contend that almost everything we know about the human brain today comes from work on other animals because of this thing called homology. We inherited features from a common ancestor. So even though I might not know the human condition, which is an unknown form, I can do this comparative analysis. And these are cartoons of brains, and each of the different colors is a different cortical field. And I can see that there's a constellation of cortical fields that all species have. And the most accurate inference I can make about the human condition is that it has these fields as well, like a V1, a V2, a primary somatosensory cortex, a second somatosensory cortex, and so on. I can also understand another unknown form, which is a common ancestor, because I could say, well, there's this pattern, a common pattern of organization that all species have. Did this evolve independently tens of thousands to millions of times in different lineages, or was it simply inherited from a common ancestor? Really interesting. you can't get rid of some aspects of cortical organization. And this is a really nice study, and this is modified from Bronstein et al. And this is a blind mole rat. So here's the nose of the blind mole rat. Here are the teeth. This is the eyelid, and the eyelid has grown over the eye, and the eyes are microphthalmic. They're about the size of a pinhead. So these animals only use vision for circadian functions. Yet they cannot get rid of a geniculocortical striate system. They still have an architectonically defined V1. It's just that it's been co-opted by the auditory system. So there are constraints in how you can build a neocortex. It's not like anything goes, and it's probably due to the contingent nature in which genes are deployed during development. If then. If you pull out a contingency that happens very early in development, you have a non-viable organism. Okay. So there's common features of organization. We can identify them in all these species, but there are also differences. So this is a flattened view of a macaque monkey to the left and a mouse to the right, and they're not drawn to scale. Rostrals to the left and medials to the top, and I can identify a primary visual cortex in blue here, and I can identify that same cortical field in mice. I can identify a somatosensory cortical field in red, an auditory cortical field in yellow, and but I can see that there are large differences. There are changes in the relative size of these cortical fields that clearly changes in the number of cortical fields. And I'll go through these in the next slide. But if I did, this mouse is not drawn to scale. And if I did draw this mouse to scale, I want people to appreciate that the mouse cortex is very, very small compared to a macaque monkey cortex and infinitesimally small compared to a human neocortex. Okay, so I want to talk about systems level modifications that have been made to the brain. And we came up with these modifications by doing a comparative analysis in a lot of different species. And I think I've worked on maybe 45 different species of mammals to date. So these boxes here are meant to indicate the cortical sheet. So these are some of the systems level changes that have been identified in different mammalian species. So you can have a relative or an absolute change in the size of the cortical sheet. You can have a change in what we call sensory domain allocation, and that's the amount of cortex that's devoted to processing input from a particular sensory system. So auditory is in yellow here, visual is shown in blue, somatosensory is shown in red. I can have, so some cortices can have a lot of cortex devoted to visual processing, and some cortices can have a lot of cortex devoted to auditory processing in an echolocating bat, for example. You can have a change in the relative size of cortical fields. You can have a change in the magnification of behaviorally relevant sensory surfaces. And here I have behaviorally relevant body parts for the somatosensory system. So, you know, if I'm a human, I have a huge representation of my hands and my oral structures and supralaryngeal tract. If I'm a rat, I have a huge representation of my vibrissae. We can have a change in the number of cortical fields, which I introduced initially in this talk. And we can have a change in the connections of cortical fields and even a change in the connections of what are homologous cortical fields. So what I want to talk about today are sensory domain allocation, magnification of behaviorally relevant body parts, and changes in cortical connections, and how these modifications arise. So what factors contribute to these systems level changes? What is the time courses over which these changes occur? Do they occur only over large evolutionary timescales, or can they be induced rather rapidly? And so so I'll talk a little bit about our comparative work, but I'm also going to talk a lot about our developmental work. And what we're trying to do with our developmental work is to see if we can induce those sort of systems level modifications in a developing nervous system and generate a phenotype that is consistent with what evolution would produce. Okay, so I'm going to give you the conclusion of my talk, and then you can listen to the rest of my talk. I show really cool pictures of animals and stuff, so you might want to stay, or you can go away and say, okay, I know what the talk's about. But here are the conclusions. There is no single way in which a change occurs. I mean, when I was first starting out as a graduate student, I would think of what is the way you increase the number of cortical fields? What is the way in which you can increase the size of a cortical sheet? There are multiple mechanisms that generate similar phenotypic outcomes. And any animal is a combination of different factors that contribute to the phenotype of the brain, the body, and behavior, combination, hence my title. And changes occur over two timescales, over the long evolutionary timescale, and over shorter developmental timescales and within the life of an individual, and even over just a couple generations. Okay, this is the outline of my talk. I'm going to give you examples from the natural world, differences in peripheral morphology and use. I'm going to talk about some experimental manipulations in peripheral morphology that we've made in an animal model, a new project called Laboratory Rats Gone Wild, and then I want to talk about natural variation in early experience within a population. Okay, changes in peripheral morphology and use, and this is just one of my favorite comparative studies I've ever done. And this is a duckbill platypus. I went to Australia to work on duckbill platypus. And I'd like to say, I had to catch all the animals I worked on when I went to Australia. Prior to that, I was just working on brains in a laboratory. And so catching these animals and, experiencing them in their natural habitat made me realize that you can't just look at the brain in isolation. You actually have to think about these brain-body environmental interactions. So the duckbill platypus is an extreme example of morphological specialization. So this is the bill, and there are stripes of mechanosensory receptors on its bill, and they are interdigitated with stripes of electrosensory receptors. And when the animal's doing anything important like navigation, catching prey, or mating, it closes its eyes, ears, and its nose, and it's one big, huge bill moving through the water, living its life. Okay. So if I record from the neocortex and I put thousands of recording sites and map out the body of the representation of the body in somatosensory cortex, this is what I come up with. This is a flattened view, rostrals to the right, medials to the top of the neocortex. And this is the platypunculus. And so this big light blue representation is the primary somatosensory area. The tail is right here. The representation of the hind limb is here, the forelimb. Basically, the body is really, really tiny. And there's a massive representation of the bill. And the blue are mechanosensory receptors and the red are electrosensory receptors. So about 90% of the entire S1 is devoted to processing inputs from the bill. There's another rostral somatosensory field that has a huge representation of the bill, and a caudal somatosensory area that has a large representation of the bill as well. Here's auditory cortex embedded within somatosensory cortex and the primary visual cortex. About 75% of the entire cortical sheet is devoted to processing inputs from the bill. So this is extraordinary. So of course, here are the questions. To what extent are these differences in the cortex associated with genes intrinsic to the neocortex that are altering over the course of evolution, genes associated with the development of the body that are altering over the course of evolution? Because remember, you've got this big, huge bill, but you also have this bill that in the peripheral morphology that engages in these specialized behaviors. What about epigenetic influence on body morphology, like diet and gravitational stress? Sensory stimuli present throughout development and throughout the adult lifetime, the use of that particular body part, and the affordances in the environment. So we know that genes intrinsic to neocortex alter cortical field size. And this is a study by Zembricki. And what he did was they overexpressed a transcription factor called EMX2. And this is very important in cortical development or in the process of arealization or the emergence of these different cortical fields in the neocortex. And what he found is that when you overexpress EMX2, you can generate one of those systems level changes that I described in the very beginning of this talk. You can increase the size of a cortical field. And so rostrals to the top here and medials to the left. Here is V1. And we're looking at this with serotonin stain, and this is V1 in an animal where EMX2 is overexpressed. And you can see it's quite a bit bigger, and they measured it, and here you go. It comprises a larger percentage of the entire cortical sheet. And if you look at the cartoons here I'm showing you this smaller V1, and some of the cortical fields appear to be pushed a little bit rostrally. But equally important, genes that are involved in the development of the body can also be tweaked during evolution. And those tweaks can generate something really magnificent, like the wing of a bat. So this is body morphology of a mouse to the left and the bat to the right. And you can see that the structure of the forelimb is really different. I can still identify a distal forelimb, a digit 5, a digit 4, a digit 3, a digit 2, and a digit 1. I have these interdigit membranes. There are a number of other specializations associated with flight. And it turns out there are only a handful of genes that are whose expression pattern is being slightly altered or that are being inhibited, that generate something from that's like a small forepod to something that's this beautiful wing. And one of those is PRX1, and this is involved in the elongation of the forearm, as you can see here in the bat. And if you look at middle stages of limb development, you can see that the expression pattern is quite a bit larger, and it can account for this change in morphology of the limb. So then if you look at the neocortex and you look at the primary somatosensory area and you say, okay, what does the representation of the limb look like in a mouse compared to a bat? And what you find is a really tiny representation of the limb, the forelimb in a mouse compared to a bat. And it's extraordinarily large. And so you say to yourself, is this due to genes intrinsic to the neocortex, or is it simply due to genes extrinsic to the neocortex, but that are involved in the development of the body that, of course, then manifests as this large representation in the cortex, or is it some combination of both? That I would argue is probably some combination of both. And we have to remember that different morphologies or morphological specializations are probably, and their representation in the cortex are probably some combination of a variety of different things, but it's not going to be the same thing in a bat versus a human, for example, with the human hand, because there's like, I don't know, like I think 100,000 years of independent evolution of those lineages. Okay, so all brains follow the same rules of construction and operate under the same constraints. And I've shown you an example of the bill of the platypus, beautiful examples from Ken Catania's lab, the nose of a star-nosed mole, the incisors of a naked mole rat. But maybe we should start thinking about the human supralaryngeal tract and oral structures in the same light. I'm not suggesting that what you want to call Broca's area isn't a behavioral specialization of humans, but it's certainly following the same rules of construction. So we might want to consider it as a magnification of these structures, these oral structures and supralaryngeal tract in somatosensory cortex, motor cortex, and premotor cortex. And we can call it Broca's area, but it hasn't been plopped into humans de novo. And there are probably changes in connectivity, amongst other things, but it is still following these same rules of construction. Okay, now I want to talk about experimental manipulations of peripheral morphology. And the animal that we use is the Monodelphis domestica, or the short-tailed opossum. We have a really cool colony in our laboratory. And these animals, what we want to do is we want to change the ratio of incoming sensory input. These are highly visual animals, and we want to completely remove it before the nervous system has developed. So the reason we do this, and the reason I show you the platypus first, is that you have this huge change in ratio of incoming sensory inputs into this naturally evolved platypus, where it's basically all about the bill. So we're trying to do the same kind of thing and make the same kind of tweak in a developing animal. And these animals have a beautiful visual system, rostral to the right, medials to the top, and this figure at the left. And this is a flattened neocortex. You can see this big, beautiful V1. You don't have to be a neuroanatomist to say, ooh, I can really see that. A much smaller S1 and A1. But here's the cool thing down here. The retinal ganglion cells have not reached the thalamus. In fact, they're just about at the optic chiasm. The thalamocortical afferents have not come near the neocortex. They're only in the internal capsule. In fact, this is what these little guys look like at the time we do these enucleations. They're really small. They have just a thin layer of eyes, and it takes about 5 minutes to do both of these to bilaterally enucleate these animals. And then we let these animals grow up and see what happens, right? It's a great experiment. I'm going to poke the frog and see what happens. Okay, so the first thing we did is we measured the size of cortical fields in these histologically processed tissues. So we say, what is the relative size of S1 compared to the relative size of V1 compared to S1 compared to A1 in these bilateral enucleated animals. So what's really important is, first of all, there's no change in the overall size of the cortical sheet in bilateral enucleates compared to sighted animals. But we do have a change in the size of V1. It gets a lot smaller, and S1 gets a lot bigger, not too different than those Imbricky studies that I showed you where we manipulated those transcription factors in the cortex, and not too different than the naked mole rat. And I'm going to show you why. So we can't get rid of a V1, and even though functionally I'll show you that it's really not being used for vision. They have no eyes. But we maintain this constellation of cortical fields, again, indicating that there are really serious constraints in how you can build a neocortex. Okay, so we assess the functional organization of cortex in adults, and what we do is we do electrophysiologic recording studies. This is a little bit different of a view than that platypunculus I showed you, but this is how that platypunculus map was obtained. Each one of these dots is an electrode site that we put into the brain, and then we recorded neural responses. And the blue dots are neurons that responded to visual stimulation. The red dots are neurons that responded to somatosensory stimulation. And the green dots are neurons that responded to auditory stimulation. And this is a sighted animal here at the top left. And this is a bilateral enucleate at the bottom right. And what you can see in what would be what would be visual cortex, the neurons have been, that cortex has been co-opted by the somatosensory and auditory system. Again, kind of similar to our naked mole rat. Did we produce something that is consistent with what evolution would produce? And in some ways we have. We couldn't get rid of a V1. We can measure it architectonically, but the system has been co-opted. Here's what's really cool. If you look at receptive fields in that reorganized V1, you see that they're predominantly on the vibrissae, the snout, the nose, and the face. And so in essence, we have this huge representation of this now behaviorally relevant body part. And I'll show you this in our behavioral studies in Monodelphis domestica. So we we've kind of generated something a little bit like the platypus, where you just have this massive representation of these important body parts. And of course, these body parts are used in important ways. Okay, so now what about connections? So what we did is we put anatomical tracers into V1, or the reorganized V1 in bilateral nucleates. And the top are normal connections. And this is just a graphic illustration showing those. And the blue, all the blue areas in the cortex and thalamus are represent, visual areas versus auditory in yellow, somatosensory in this orange color, and multimodal in green. And you can see that V1 in a sighted animal predominantly gets input from visual areas of the cortex and visual nuclei of the thalamus. You have a different story here in P4 enucleates. And what you see is the black arrows are connections that are the same. There might be differences in densities, but again, it's really constrained. You cannot get rid of these patterns of connections. You still have a little teeny LGN, and it still projects to that architectonically defined V1. The red are new projections or novel projections that you don't see in normal animals. So you have projections from somatosensory areas of the cortex, somatosensory areas of the thalamus in orange here, auditory areas of the thalamus and auditory regions of the cortex, and differences in other connections as well. Okay, so this is really important because a lot of times when we do these kinds of studies, the only thing we're looking at is the targeted system, the visual system. But changes are not restricted to the visual system. So what we did is we put anatomical tracers into the primary somatosensory cortex in sighted animals and P4 enucleates. This circle right here is the center of an injection of an anatomical tracer, and this is a reconstruction. And these little tiny dots are retrogradely labeled neurons, which means this area FM is projecting to this portion of S1. S2, these little cells in S2, they're projecting to S1. And so you don't have to be, again, a neuroanatomist. This is just a simple visual matching task. You can look at this and say, okay, this is getting, S1 is predominantly getting projections from other somatosensory regions. But there's really, really widespread projection pattern in the bilateral enucleates. And we didn't just eyeball this and say, ooh, this looks really different, although it does look really different. We actually, down here, if you want to, you don't have to take my word for it, this is published, we actually quantified those differences in connections. And then we also looked at the vibrissae representation in primary somatosensory cortex and recorded from single neurons there and looked at receptive field size and neural response property. And what we found was that these early blind animals had much smaller receptive fields in the vibrissae representation, for neurons in the vibrissae representations. And those neurons had better discriminability. And what I mean by that is that this is the snout of a bilateral nucleate, of a Monodelphis domestica, and these are rows of whiskers and then arcs of whiskers. And by that better discriminability, if I could say it, it's getting towards the end of the day here, it means that those neurons are able to discriminate stimulation of this whisker compared to surrounding whiskers in a bilateral nucleate compared to a normal animal. But what about behavior? And so we started doing behavioral studies about 5 years ago in my laboratory, and man, they were the hardest studies we ever did. But it's really important, I think, to do this because we show all these cool changes in the cortex in functional organization, neural response properties, cortical connections, but those are not the target of selection. Behavior is the target of selection. External phenotype is the target of selection. Genes are not the target of selection. They may co-vary with body type. They may co-vary with cortical connectivity or co-vary with the size of cortical fields, but they are not the direct targets of selection. Okay, so before I go to the behavior part, I want to say something important, and that is that I'm sure the whole world knows this. All these human researchers know that short-tailed opossums, they whisk, like commonly used murine rodents, which means they use their whiskers in important ways to explore their environment, palpate objects, to determine the size and the texture of objects, much similar in some ways to the way we use our hands. And so I just want to show you, because it's fun to show movies. There's a sighted animal, there's a bilateral nucleate animal, they're both whisking. But I think what's more telling is that activity levels in sighted and bilateral animals are the same. So this is a pretty cool slide. And this is kind of sped up a little bit. They're really not this fast, but they are kind of fast. And sighted animals, we put a blue circle over sighted animals and a red circle over bilateral nucleates. And this is a different set of experiments where we're trying to enhance their environment. And there's all kinds of trees and things to climb on, and we change them out every 3 days so that when they navigate this space. It's not a memory task or spatial memory task, but they have to actively use their sensory systems to figure out what's going on. Their nest boxes are up here and food and water are down here and toys. And so we kind of we change this and reconfigure it every 3 days. So check this out. This is a bilateral nucleate up here. He's up at his nest box. Bam, he just comes right down this thing. And you're going to see him. It's really cute. They have prehensile tails, and he's going to be gathering all this nesting material, and he's putting it in his tail. And he's doing all this here, and he's going to go right back up here with, he's not falling off and acting like he's in any way impaired. In fact, if I didn't point this out to you, you would have a hard time figuring out which is the normal animal right here and which is the bilateral nucleate right there. Pretty cool. Okay, so these are the behavioral experiments. This is the variable rung, ladder rung walking task. Should have practiced that. And here's what you do. It's pretty cool because you don't have to, you train the animals to do this, but they do this really rapidly. So they have a box at this end and their home cage, it's hard to do this on Zoom, their home cage is at this end. And they're in this box, and strung between these 2 boxes is a ladder. And there are plexiglass on each side. And this ladder is suspended fairly high off the floor, so they don't want to fall. And so initially these rungs are equidistant. And so they really want to get out of this strange box that they don't know, and they want to run across to their own home cage. So that you can train them to do that pretty rapidly. And what's nice is this is a naturalistic task. Then what you do is once they're doing that, you randomly change the distance between the rungs so that now it's not equidistant. It's, a wider distance and shorter distance and wider and shorter and so on. And they don't have any practice with that. And so what's really cool is we're using Deep Lab Cut, which is a great method in which it's a machine learning algorithm that allows you to not just look at success rate, how well did they do or how many errors did they make, but you can actually look at the kinematics. So the user places these little balls on the area of interest or the body part of interest, and you can see what happens to the forelimb when it slips and what happens to the tail when the forelimb falls through those rungs. So what I'm going to show you is sighted and blind animals navigating this task. And this is really beautiful results. I know it's my own laboratory, but it's okay to think that your laboratory is doing really beautiful stuff. Okay, so this is a sighted animal walking across and he has whiskers, and you can see he makes errors when he falls through, bam, he falls through. So you can count those errors, and you can see what happens to the limbs and the tail and the nose and everything when he's making those errors. So check this out. This is an early blind animal, and they are a lot better. I'll show you the statistics for that. You don't have to take my word for it. And they're doing pretty well. But this is the really cool one. This is what happens after we trim those whiskers. And now let me remind you, remember that big, huge whisker representation in the reorganized V1? Remember the changes in the response properties of neurons in the whisker representation of S1. There are changes in connections of S1, changes in connections of visual cortex, and maybe they're doing something, and maybe it has to do with the whiskers, because, okay, this is that same animal. Oops, I kind of ruined that exciting part. All right, this is the same animal after we've cut its whiskers, and you can see that it completely falls apart. So maybe those changes in the cortex and thalamus are doing something. And we think it's that in fact these animals are using the information from the whiskers in a very different way than sighted animals to do these sorts of navigation tasks. And this is just, we've actually quantified these differences, and you can look at total error. And this is so if these histograms, if these bars are smaller, the animal does better, makes many fewer errors, and you can see it's quite different than sighted animals. And if we trim the whiskers, both of these animals do poorly. But what's really interesting, if you look at the whisker trimmed animal here, it does about the same as the animal with whiskers here. So the whiskers are really important in these guys. And what's really interesting is that look at this picture down here. Here's a normal postured bilateral nucleate crossing the ladder rung walking task. And here's what happens when the whiskers are trimmed. They have a complete change in the posture. They don't lift their paws high. Their stride isn't as long. They tap their nose on the ladder more often. Both do. Their tail height changes, so everything changes. And the sighted animals are affected by this as well, but not to the same extent as the blind animals. And so it's sort of like walking across the room and then all of a sudden somebody turns the lights off and kind of start doing this much more conservative locomotion. And then this is just a tactile discrimination task. And the animals were trained to discriminate between a smooth surface and some grit of sandpaper. And bilateral nucleates, in this case, this is difference in sandpaper on this axis, and this is accuracy or percent correct. And so if there's no difference in sandpaper, you just fall to chance, right? They're both smooth or they're both really rough. This is a large difference in sandpaper, versus, like smooth versus a really heavy grit of sandpaper. And as you move in this direction, the task is still pretty hard, and it gets easier and easier and easier. And the bilateral nucleates do this tactile discrimination task significantly better than the sighted animals. And in fact, they can discriminate differences in grit of sandpaper of 25 microns, whereas sighted animals need at least 100 microns to be able to tell the difference between different grits of sandpaper. So they're doing better with these in these tactile discriminations. And once again, when we trim the whiskers right here, this is the mestacial whiskers, both animals fall apart, but then after a few days they get better. Because they actually changed their strategy, but I don't have time to talk about that. And then if you trim the genial whiskers, only the bilateral nucleates fall apart. Okay, so to end this little segment here, I want to say that blindness is more than an absence of light. You can't just close your eyes and say, oh, I know what it's like to be blind. You do not know what it's like to be blind, because your brain is not hardwired the same way if you had this major loss of sensory input throughout development. The functional properties of your neurons are different, your receptive field sizes are different, your cortical connections are different, and you kind of get the idea. And ultimately, your behavior is different as well. Okay, so now, what I just showed you was taking a sledgehammer to the system. There's nothing wrong with experiments to take a sledgehammer to the system, because they help us uncover sort of features of change that we might not be able to detect if we did something very, very subtle to the system. But this is a more subtle set of experiments. These are natural variation in movement and experience and affordances, laboratory routes gone wild. I made this joke. Todd, Black Square guy here, who's the technical guy who's been helping me out a lot, he's heard this talk already, because I gave this talk last week and there were some technical difficulties, and I'm hoping this talk is going to be as good as the next one. So now I'm really going to go out and hang out with my friends Cindy and Rob and my husband Scott, and this will be me tonight. I'm not going to be the designated driver, because this is the second and very last time I'm giving this talk. Okay, so these are really cool experiments because I don't have any skin in the game, and I'll tell you why. So what we do, well, first of all, let me just ask this question. If you're rearing an animal in a cage like this, can it really exhibit species typical behavior? I would say no, not really. It'd be like rearing a human in a cage. So what we do is we take advantage of these field pens that we have in Davis, and they're about 3,000 times the size of a normal cage. So this isn't just like kind of environmental enrichment where you're twice as large as a regular cage. We take pregnant female rats, and we put them out in those field pens, and we fill the field pens with all kinds of cool stuff and things to climb on and objects to discriminate, and we switch those around about once a week, and we let them give birth out there, and then we look at their young. We also look at the mothers, but we look at the young, but they have access to wild predator calls, little bugs that get into their cage. We feed them and water them, and we have to go out there every day, and it's a lot of work for the graduate students and postdocs to work on this project, but it's really exciting because given what I just showed you about, in these opossum experiments, I'm expecting a lot of things to look different across the cortical sheet. There's a range of environmental conditions. The temperatures range from 43 degrees to 12 degrees Celsius. So they're out there in the summer, they're out there in the winter. We even had an earthquake out there. Well, it wasn't just at the field pens, it was in all of Davis. As I said, they have access to predator cues, wild turkeys, raptors. And this is, I'm pointing to my screen. Let me do this. This is just an empty cage, but like I said, we fill it with different trees and things to crawl on, and it's kind of exciting. Okay, now I'll give you an idea of what they're doing out here. Let's see. So I want you to look at their tails, their trunks, their hind limbs, their forelimbs. They're crawling around a lot. Look, even when it's drinking water, it's the position in which it drinks water. So there are a lot of different affordances that a cage, just a laboratory small cage, does not have. And of course, I can go on about their huddling behavior, their playing behavior. So, we chart their progress. And so, for example, eye opening, when do they open their eyes? And so this is what's so fun. Everything we look at is different. So the field pens, this is in green, they open their eyes later than laboratory animals. They start walking sooner than laboratory animals. What we wanted to do And of course, we want to look at visual cortex and somatosensory cortex, but we started by looking at motor cortex because we thought, the affordances are so different. The synergies that they're using, the behaviors are way different than a laboratory animal. So this is different than the electrophysiological recording experiments. What we do is we insert a stimulating electrode in hundreds of sites in the cortex, and we pass a small current, and we look at the animal and see which body parts moved. They're actually kind of fun. These animals are anesthetized. We put them in a special sling, and so these are kind of hard to do. So each one of these white dots here is a stimulating electrode site, and then these colors represent which body part moved. So if I'm in this green area right here, it means we can evoke a movement of the wrist. What you'll notice when I show you the big maps is that that most of these squared areas are striped because we very rarely just have a single muscle movement. What we'll see sometimes is something like running. You can see something that almost looks like a hand to mouth. You can see what looks like a grasp. So it's groups of muscles are involved in these movements. I'm not saying that we're evoking behaviors, but we're evoking movements that are parts of probably complex behaviors. And so if you look at the two maps, the left is a laboratory animal and the right is a field pen animal. And you can see these nice maps of the cortex. You can see some differences, I think, in these forepaw representations, and certainly in the hindpaw representations, where we have a lot more hindpaw. We have some hindpaw in isolation. We also have tail. We can evoke tail movements, which we can't evoke in the laboratory animal. But this is a cooler type of analysis. And what this is showing you is movement combinations that we evoked in a laboratory in green versus laboratory, this is my second time doing this talk, a laboratory in blue and a field pen animal in green. And you can say how this is the percent of all movements where the Vibrician forelimb, where we evoke Vibrician forelimb movements versus Vibrician hindlimb. What's really cool is this part over here, where it's just all green, where we evoked forelimb and hindlimb in laboratory animals and didn't evoke that in field pen animals and didn't evoke that in laboratory animals. Forelimb and tail in field pen animals, but not in laboratory animals. Hindlimb and tail. So you kind of get the idea. These animals are using those body parts in a very different way. And this is manifest as this representation of these synergies in motor cortex. But there's evidence in other mammals that maps are built during development. So that's what I'm contending, that these motor maps are built during development based on commonly used synergies. And it looks like, for example, in the laboratory animal, because we're only evoking lots of movements of the forelimb, vibrissae, and face together, the animal's literally truncated. Its motor map is truncated. So there is evidence in other mammals that maps are built during development. And this is a recent study in our laboratory where we did the same sort of thing, where we were doing these intercortical microstimulation maps and hundreds of sites, I'm across the cortex, across motor cortex, somatosensory cortex, posterior parietal cortex. And I'm not going to show you the whole maps. What I'm going to show you, though, are sites where we were able to elicit different types of precision grips. And a precision grip is when you take your opposable thumb and oppose it to different digits, not just this, but it could be this, digits to the all digits to the thumb, for example, right? So this would be sort of, for example, flexion of D1 to D2. This would be the baseline movement, and then when we would pass a current, we would get something that looked like this. Okay. And so what I want to show you here, so there's a whole bunch of these types of really cool, complicated, fine control of the digit that are represented in cortex. And the blue is extension of 2 digits. The purple is mix of flexion and extension involving more than 2 digits. The point here, and these are 2 different macaque monkeys, and rostrals to the left and medials to the top, is that if you look at the pattern of these colors in this animal versus this animal, they're quite different. And so some of this might be due to experimental error. I don't think a lot of it's due to experimental error, but I think a lot of it's just due to variability in terms of what these animals were doing with their hands during development throughout the course of their life. And you have to remember at Davis, these animals get, and I'm sure other places as well, but they get a lot of enrichment. They got a lot of different toys. So their early experiences were probably quite a bit different. This is a really great study. This is by Shrima Mitt, and this is in humans. And what she showed is that she looked at individuals that were born without arms, and she looked at how they use tools. And most of them, these are different subjects here, D1, D2, D3, D4, they're all different subjects. And you can see the orange is lower limbs. And when they were engaged in using tools, almost all of them were using their feet. And if you've seen some of these videos of these people online, they're really extraordinary. They can play the guitar, they can paint, they can do a lot of dexterous things with their digits that I couldn't even dream of doing, or nor could you. What's really nice, this is an fMRI activation, and if you look at the cortex that's activated with their foot, these animals, well, humans are animals actually. These humans have no forelimbs, and these humans have no forelimbs. And I want you to concentrate on posterior parietal cortex. And normally, what you're going to see in posterior parietal cortex is just a boatload of activation by the hands, because posterior parietal cortex is about hands and planning movements of the hands and the eyes to explore the world, right? And what you see here is a preponderance of a representation of feet. And so this cortex is clearly, it's not based on a effector that's genetically determined. It's based on what you're doing during development with what limbs and what is now your major effector that builds this cortex. Okay, but here's the big question from the laboratory rats gone wild experiments. Can rearing in a highly variable environment with a large number of affordances alter the ability of the animal to meet a wider range of sensory motor challenges? And I think so. And if so, there's probably changes in the cortex and probably throughout the nervous system, certainly at a synaptic level. So if you think about a laboratory animal, or even a human reared in a really small important, the affordances are not high. And the variability within the environment may not be very high. So the lights go on and off at the same time every day. The temperature is the same. They get their cages changed the same time every week. So there's the distribution of the variability in their environment is like really, really tight. Okay. So now I just want to go to the very final part of my talk, which is a much more subtle set of experiments. We take advantage of prairie voles, and you may or may not know that prairie voles are monogamous and biparental. And that means that both parents rear the young. And there are prairie voles that we can classify as high contact and low contact. So there are parents that have a lot of tactile contact with their young that we can actually measure, right? And then there are those that have low, and there's that whole distribution in between, but we're only taking those two ends of the distribution. Also, I want you to take note of this specialized perioral region that has all these hairs around on the lips, on the nares, and even in the palate. And I was thinking about this, I was kind of going through my talk again today. I have so many pictures where I have taken digital images, or in the old days, photographs of anesthetized animals and special body parts. And it is actually kind of creepy, but I have tons of them, like tongues of bats and all kinds of stuff like that. Okay, so anyway, almost done here, guys. Okay, what's really cool about this, and I want to say that high contact parents are not better parents and low contact parents are not crappy parents. It's just, this natural variability. And you can imagine any scenario where having a lot less contact would be better. But what's really cool about this is that we can actually quantify the amount of times that the parents touch the offspring. And this is such quantification. You can see it's significantly different. High contact is in red and low contact is in blue. So this is maternal contact and this is maternal and paternal contact. And you can see these differences. What's really cool is that high contact offspring become high contact parents. Low contact offspring become low contact parents. And if you cross foster them, so on the day of birth, if you take a low contact baby and put it with high contact parents, it becomes a high contact parent. So it's sort of this social transmission of parental rearing style. So I think that's super cool. So what we did then is that we said, well, let's look at somatosensory cortex, right? Because there's clearly a lot of variability, or a lot of difference, I should say, in the amount of tactile contact these animals are getting when they're young. And so we put an injection of an anatomical tracer into that perioral mouth representation, because that's where a lot of the contact is taking place, in a high contact animal and a low contact animal. And you've seen something like this before. These are the injection sites right here, and these little dots are retrogradely labeled cells. So you can say, well, these projection patterns of different cortical fields to S1 look pretty similar in the high versus low contact. But if you look carefully, you can see that there are differences in density, and some of those differences can be quite large. And we did also quantify this, but this is just an easier way to show you this. And hold on. Okay. And if I look at the interhemispheric connections, so these would be cells projecting to S1 in the opposite hemisphere, I can see quite large differences in density of connections, and then actually identify projections that exist in low contact that don't exist in high contact. So this is a lot more subtle than my bilateral enucleations and even the laboratory rats gone wild, but we're seeing these same sorts of differences based on early experience. Okay, so what factors contribute to the cortical phenotype? I'm going to sum up here right now. Genes, of course. They can control cortical sheet size, cortical field size, cortical connections, peripheral morphology. Really, really important. The cellular mechanisms involved in plasticity, which then allows the environment to actually modify these same things. And so I've given you an example of the bill of a platypus, the tongue, the larynx, the lips of a human being. But what about things like social learning, language, and culture? These are all complex patterns of physical stimuli that vary during development within the lifetime and in species throughout evolution. So I think we need to actually start thinking about our behaviors in this serious way, that they're simply patterns of complex stimuli impinging on a developing nervous system. So the love of a mother is temperature, it's touch, it's cadence of a voice. All of those complex behaviors, human behaviors, in some ways I think we can break them down and say, if they vary during the course of development, then your brain's going to change during the course of development. And it can change in ways that I just showed you from my animal experiments. So this is a little human evolutionary line here. And morphology is to the top, and environment and social context is at the bottom. And so I want to point some things out, that the human hand has been around for a very long time, but we weren't doing a whole lot with our hands, certainly not the sorts of complex manual behaviors that we engage in today, and certainly not even anything close to the tool use that we have today. Our large brains have been around for a really long time as well. So we've had these large brains and these large hands, and yet our behaviors were relatively simple and unsophisticated compared to the sorts of things we're doing today. And I'm going to cut ahead a little bit. The earliest anatomically modern humans have been around for 300,000 years. That's a really long time. And that means that, there has been no change in DNA sequence. About 300,000 years ago, we were using spears and knives. About 70,000 years ago, stone tools, perforated bones, sophisticated blades. And the Industrial Revolution happened less than 300 years ago. And yet our behavior 300 years ago, even 300 years ago, was really, really different than it was today. And if you believe that all behavior is generated by the brain, and I do, that means there must be changes in the brain. And I suspect they're the sorts of changes that I showed you in my animal experiments. And I will say that, automobiles have, if you look at, the snowball of cultural evolution in humans, it's extraordinary. I mean, automobiles, 1886, not very long ago, the World Wide Web, I realize I have a misspelling here, 1990s, and now we have daily and prolonged interactions with machines and computers. So my generation is quite different than generations before me and generations after me. And I would contend that if I were born 300,000 years ago, I would not be the same at all. My brain would be fundamentally different. I wouldn't be Leah Krubitzer, just in a different culture. And I would be a very different individual if I were born 20 years earlier or if I were born 20 years later. And I think you get that idea. So I'll just end with this. We are combinatorial creatures constructed by genes, bodies, behaviors, and environmental contexts. Humans have evolved an extraordinary capacity to construct our neocortex based over the course of a prolonged infancy and childhood, allowing for rapid phenotypic change even within a single generation. We have a remarkably fluid brain-body interface with the environment, such that tools and machines can extend our embodiment and peripersonal space and expand the loop between our brains, our bodies, and the world. This has made us into unique biohybrid creatures whose brains adapt and bootstrap themselves with the technologies they have given rise to, and with whom our futures are increasingly entwined. So our tools are not simply an extension of ourselves. They are now part of the human, I would say part of the human body, or part of the human brain-body experience. And I'd like to thank my funding agencies. And again, thank you for inviting me to give this presentation.